So I had come up to the discussion of NK cells and had explained about how the NK cells are involved in killing the cells that are infected with intracellular pathogen, that is bacteria or virus. So they are responsible for secreting that granzymes, and those granzymes are discharged into the target cell through the tubular protein called as perforin, and those granzymes get activated in the target cell, and there they bring about cytolysis or cell lysis of the target cell. We'll talk about the another cell, that is gamma delta T cell. The gamma delta T cell gets the name because of the gamma delta receptor that is present on that T cell. About this receptor, we'll talk about it in the unit 3. Now this T cell, gamma delta T cell, is restricted to epithelial lining. It is present in almost all epithelial linings. And it doesn't remain in one place, it keeps traveling. It travels on the surface of the epithelial lining, goes to the bottom of the epithelial lining, and keeps moving about by amoebic movement. As it is traveling along the epithelial lining, when it recognizes a cell which is infected with bacteria or virus, because that cell is in distress and it will release those distress proteins, one of them being heat shock protein. The gamma delta T cell gets arrested there with respect to that cell which has been infected and brings about the same mechanism as NK cell did. As soon as it gets arrested with the cell that is target cell which is infected, it undergoes activation, enlarges in size, produces those granzymes in large quantities, and produces the perforin and inserts that perforin into the wall of the target cell and releases the granzyme and kills the target cell. In other words, gamma delta T cell is also a killer cell. So from the examination point of view, if any of these two cells are asked, you are expected to draw a diagrammatic representation of the structure of the cell and its interaction with the target cell, which is shown in this slide, bottommost left-hand side. You need not show the entire sequence of events. You just show the bottommost left-hand side interaction. And labeling of the cell, you be careful. If it is NK cell, you label it as NK cell. If it is gamma delta T cell, you label it as gamma delta T. Both these two cells, even though they are two different types of cells, bring about the same type of action of cell killing when the cell is infected with intracellular pathogen. Since gamma delta T cells are restricted to the epithelial lining, they are also once in a while called as intraepithelial T cells, IET, capital I, capital ET, intraepithelial T cells. Their sole function is to patrolling the epithelial lining to see for the infection. If there is infection, they will attack it. As I had mentioned, NK cells are responsible for inflammatory response because they secrete inflammatory cytokines like interferon gamma and TNF alpha. Same type of cytokines are secreted even by the gamma delta T cells when they attack the cell that is infected with bacteria or virus. So you know now, NK cells and gamma delta T cells, which have the innate ability of attacking the cells that are infected with virus, are responsible for partly inflammatory response. And if that inflammatory response goes uncontrolled, it is detrimental to the individual. It causes a lot of damage. We'll come to that part little later. Now we will see the another type of cell, that is CD5B cell, The CD5 B cell is a special type of B cell which is having a receptor called a CD5 on its surface. In addition, it has a receptor for antigen, and that receptor for antigen is pentameric IgM molecule, immunoglobulin M molecule, pentameric. Pentameric means it has got 5 monomers joined together to form pentamer and has got ability to bind to 10 antigens at a time because it has got 10 antigen binding sites. At the same time, this cell is restricted only to peritoneal cavity. Peritoneal cavity is the cavity found in the abdominal region in which your digestive organs or digestive tract is suspended, and there is some amount of peritoneal fluid there which lubricates these organs and prevents these organs from attaching to the body wall. And in this fluid, these cells are suspended. CD5B cells are suspended. Only when the peritoneal wall is damaged due to accident and punctured, and the pathogens enter the peritoneal system, these cells comes to the rescue of the person. Otherwise, these cells are very quiet and they are restricted to only that region, and very rarely any pathogen enters the peritoneal cavity. Very rare instances. Only when mechanical injury like rupture of the abdominal wall takes place. So as soon as, let us presume that the abdominal wall is ruptured, the pathogens entered by accident, the CD5 B cells get activated because they bind to that antigen readily through their pentameric IgM molecule, and they swallow this antigen and process it and destroy it. At the same time, those cells which were bound to the antigen, CD5B cells, get activated. And those activated CD5B cells will start secreting copious amount, large amount of IgM molecule. And this IgM molecule will not be membrane bound now, it will be poured into the peritoneal cavity to bind to other antigens which are there free. So that is how the IgM molecule helps in binding to those antigens. And I had told you that IgM molecule is the antibody that is there even as our anti-A and anti-B antibody, which is responsible for agglutination. Same way, if the bacteria had entered the peritoneal cavity, this IgM molecule will bind to it and causes the agglutination of the bacteria. And agglutinated mass of bacteria is going to be lysed or destroyed. That complex is going to be destroyed through complement mediated lysis. And this process we'll talk about that when we are going to talk about classical complement pathway at the end of this unit. But for the time being, you keep in mind, when I had dealt with complement fixation assay, I had just made a reference to it. So the complements help in removal of the antigen antibody complex. Since CD5B cells are innately responsible for preventing the infection of the peritoneal cavity, and they secrete only one type of antibody molecule, that is IgM, which is temporary measure, and therefore CD5B cells do not give long time or long-term immune memory. It is a short-lived immune memory for the immediate protection. And once the bacterial infection goes, these CD5B cells become quiet and they remain quiet there. And once again, if there is an infection, they get activated. So the immune memory is not there and they respond innately. And they are restricted only to the peritoneal cavity. So we'll go to the another process, that is now inflammation. Inflammation is an innate response. An innate response shown by an individual to restrict the infection or contain the infection in one place, or to prevent the infection from spreading all over the body. And that is the reason the inflammation takes place. But it is not a pleasant feeling. When the inflammation takes place, there is going to be a lot of changes that are taking place locally in that area. And that area becomes highly inflamed and painful. The word was derived from the flame. When you touch the flame, area will burn and burning sensation will be painful. Same way, the inflammation is a process where it causes the painful response. What are the things that happen when there is inflammation? Let us take the example of wound. Okay, let us presume that to the extreme right of the slide, you can see there is a wound that has taken place. The skin is ruptured and the bacteria have entered through that wound. Skin is ruptured and the bacteria have entered through that wound. As soon as the bacteria enters that place, your body responds immediately. And to that area, large amount of blood is going to be pumped. Therefore, you need vasodilation. Vasodilation means enlarged blood vessels. Girth or dimension of blood vessel increases. A large amount of blood comes to that area. Why large amount of blood comes to that area? Because it has to bring all the defense cells into that area. As a result, that area gets red in color and the process is called as erythema. 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 Dilation of blood vessels results in erythema. Second, those dilated blood vessels become increasingly permeable for the plasma to ooze out. The plasma that oozes out will release lot of bacteriostatic agents. Even defensins will be released and complements will be released in that area. And that process of increased plasma leakage results in swelling of that area. You have all experienced when you are wounded. The wounded area becomes red in color. Around it, there will be redness. And then the wound becomes swollen and highly painful. You can't touch that wound for some time. And the swelling of the wounded area is called as edema. Edema refers to the accumulation of fluid in that area. And the fluid is nothing but the leaked plasma. And sometimes that leaked plasma also oozes out of your wound in addition to blood. For some time, when the wound has taken place, initially the blood will come. After that, the blood will clot. And once the blood clots, it forms a scab over your wound as a protective layer. And then the plasma will be oozing out from that area. At the same time, when these two processes have taken place, because of the vasodilation, you will see that large number of neutrophils will have come to that area. And those neutrophils will get a special ability. And that special ability is to come and attach to the wall of the blood vessel. Otherwise, they are free. They do not attach to the wall of the blood vessel. They are free moving cells. But only during the process of inflammation, they come and attach to the wall of the blood vessel. And that process is called as margination. Margination is a process where neutrophils will come and attach to the wall of the blood vessel from inside. Next, The process will be those neutrophils will squeeze out of those blood vessels, which is a special ability, squeeze out of the blood vessel into the area where the bacterial invasion has taken place. They will, in large numbers, they start squeezing out. And that process of squeezing out, which is a special ability of the neutrophils, which happens during inflammation, is called as extravasation. Getting out of the blood vessel is called as extravasation. The other word for that is diapedesis. Diapedesis or extravasation of the phagocytes. Phagocytes are nothing but the neutrophils. Is a special ability that is gained by the neutrophils during inflammation. And large number of neutrophils flood that area where the bacterial invasion has taken place and start swallowing those bacteria. In the process, some neutrophils will die because bacteria has already released some toxins. And those toxins are harmful to the neutrophils, and some neutrophils, on encountering those toxins, exotoxins released by the bacteria will. And that is why in a wounded area, neutrophils will constitute the pus. The pus is nothing but dead neutrophils. Almost all wounded individuals will develop pus in the area where the infection has taken place. It's a good sign that the pus has taken place, means the neutrophils have come there and they have defended you. But at the same time, all neutrophils that infiltrate that area where the wound has taken place will not die, because as the dying neutrophils release some chemokines, those warning chemokines will warn the incoming neutrophils to be prepared for the type of toxin released by the bacteria so that they are well guarded against the toxin. So initially there will be large number of neutrophils die, but later the neutrophils do not die. They will succeed in containing the infection in that area. Whatever the bacteria that had invaded that area will remain there only. It will not be allowed to spread. It will be swallowed, phagocytosed. With the generation of reactive oxygen species, those bacteria are going to be destroyed in the vacuole, that is phagosome of the neutrophil. Other details about the neutrophil property we will talk when we are going to talk about the cells of the immune system. For the time being, keep in mind that neutrophils are the cells that are the first arrivals when there is inflammation. Okay. The question will be, is there a necessity that always for the inflammation there is a need of wound? No, there is no need of wound. Wound is one of the characteristics. That is, skin is ruptured. There is always a possibility that the bacteria might have invaded your system and gone into your blood and gone into the tissue spaces. When it has gone into the tissue spaces, there also the inflammation is possible. Wherever in the tissue spaces, if the infection has taken place, immediately the inflammatory response happens there. Why? Because there is no tissue without blood vessel supply. Every part of the tissue has got blood vessels. As soon as the pathogen makes its presence there, the blood vessel will start dilating and start pushing out those neutrophils. And what makes the area where the invasion has taken place to be recognized is because of some receptors that are there, which are called as TLRs, toll-like receptors. We'll come to that receptor a little later. But for the time being, keep in mind that any foreign invasion that has taken place in the body that causes the inflammation in that area. And internal in situ in situ inflammation in any organ will give rise to some discomfort. Let me take an example. When you get stomach infection or intestinal infection, you experience severe cramps and pain. That is either stomach pain or intestinal pain. And that pain is the symptom of inflammation that is taking place in that area because of the infection. And you experience this pain in the stomach or in the intestine only when you have the stomach infection or intestinal infection by extraneous bacteria and you have stomach upset, indigestion. Only when you have indigestion, that time you have experienced these pains in the stomach area or intestinal area. Severe cramp will be there, pain will be there. And you know, when you are wounded, the wounded area becomes highly painful, and few days you will not be able to touch it. You may be able to touch it very gently, dress it very gently, but you will not be able to touch it very hard. You will wince in pain. The pain is because of the inflammation. So please remember, the inflammation is an innate response to contain the infection. And this inflammation is also taking place in people who are infected with coronavirus. When the virus reaches the lungs, in the lung epithelial area, they will have invaded the cells through ACE receptor. And when they have invaded the cells through ACE receptor and causing infection there is intracellular infection. Natural killer cells, gamma delta T cells have been already activated. At the same time, the alveolar area where blood supply is present is now triggered, and there the inflammation has started. And that inflammation in that area makes a lot of fluid to accumulate, and that is what it is said that lungs get flooded in coronavirus patients. In addition, there is other mechanisms which are happening, and the bradykinin is responsible for secretion of too much of hyaluronic acid, which is going to be mucilaginous, and that clogs your lung. So whatever it may be, so inflammation process is a common feature of innate immune response. Okay, then comes the mast cells. I kept referring to these cells earlier also. And what are these mast cells? They get their name from their histological architecture, mastalon. Mastalon in Greek means granules. You see the number of granules in its cytoplasm. Large number of granules are there in the cytoplasm which are stained. That is the characteristic of mast cells. And these granules are nothing but stored pharmacological mediators. And one of the major mediator of the mast cell pharmacological mediator is the histamine. Okay, histamine. And that is the one which is released in you people, which causes the effect of blood vessel dilation in some areas, smooth muscle contraction in some areas. And therefore, antihistaminic tablets are administered to the people who have very active mast cells. Now, we'll come to these mast cells. I had said that they are present in the lining of the blood vessels, and you know that blood vessels are there everywhere in our body. And since these mast cells are present in the endothelial lining of the blood vessels, they have a chance of getting exposed to the antigen that enters the blood vessel. And they are majorly responsible for allergic reaction, and they are classified under the hypersensitivity, which we'll be talking about that in the unit 4. But for the time being today, we'll talk about their response in the innate immune system. When the pathogen makes its presence in the blood, the mast cells will come in contact with these pathogens through the TLR. I had mentioned that the TLR is nothing but toll-like receptor on the membrane. And that toll-like receptor which recognizes this pathogen binds to it and activates the mast cells, and those mast cells which are activated because they are bound to the pathogen are going to release the pharmacological mediators which are already stored in their granules. And what are those pharmacological mediators released by the mast cells? They are here listed here. One is histamine, another is prostaglandin, bradykinin, leukotrienes, and large number of proteases and inflammatory cytokines like interferon gamma and TNF alpha. One more pharmacological mediator I forgot to mention here, that is 5-hydroxytryptamine. All these pharmacological mediators, when released in large quantities, they have adverse effect in the body. They cause constriction of smooth muscles, dilation of blood vessels, and the pharmacological mediators like inflammatory cytokines which are released by the mast cells cause inflammation. So you will have now in the area where there is inflammation, 3 different cells causing inflammation, and all of them will be releasing inflammatory cytokines. Imagine the amount of the cytokine that is going to flood and going to be circulated in the body. So if it goes beyond control, it is very, very dangerous to the individual. Therefore, they are always talking about cytokine storm, and you have to control it through medication. And there are medications available which in fact prevent the release of these cytokines, which prevent the release of these other pharmacological mediators like bradykinin and prostaglandins and histamines. On appropriate medication, you can control them and make them release in a smaller quantity, which is sufficient to give you protection. And that protection will be sufficient to contain the infection and prevent the infection from spreading. Then comes the mechanism of innate recognition. We kept on talking about innate immune mechanism, innate immune cells. How do these cells recognize the foreign antigen that enters the body? They have molecules on their surface, and those molecules which I told you are called as toll-like receptors, which are called as pattern recognition receptors, PRRs. Almost all immune cells in our body are provided with toll-like receptors. Then each pathogen that enters our body has its fingerprint molecule, which is its trademark. And that fingerprint molecule, which is the trademark of the pathogen, is called as pathogen associated molecular pattern. Pathogen associated molecular patterns. PAMPS refers to the molecules associated with the pathogen. PRRs refers to the defense cells. Almost all our defense cells have these PRRs on their surface. Now, what are these PRRs? They are classified into TLR1 to TLR9. Still, the functions of some of them is not yet known. A lot of work is going on. But functions of some is known, which is represented here. TLR2 and TLR4, sorry, it is spelled as TLE, it is TLR4. TLR2 and TLR4 respond to lipopolysaccharides, LPS, capital LPS, refers to lipopolysaccharide of the bacteria, and peptidoglycan layer of the bacteria, that is the capsule of the bacteria. Lipopolysaccharide and peptidoglycan are the characteristic of bacteria, and that is recognized by these two TLRs, TLR2 and TLR4. TLR7 and 9 is responsible for recognition of nucleic acids of any type of bacterial and viral origin. TLR7 and 9 meant for recognition of bacterial and viral nucleic acids. TLR3 is responsible for recognition of the single-stranded RNA. There are viruses which are single-stranded viruses, RNA viruses, and there are double-stranded RNA viruses also. And TLR3 is meant specifically for recognition of single-stranded and double-stranded RNA of the viruses only, not of the bacteria. Okay, and TLR5 is responsible for recognition of the bacterial flagellin, which is a protein restricted to the flagella of the bacteria. And there are many bacteria which are motile using their flagella. And that flagella has flagellin protein, which is characteristic of that bacteria, which is recognized by TLR5. So you know now different types of toll-like receptors that are present on our immune cells are capable of recognizing the pathogen. The pathogen is having pathogen-associated molecular pattern. And I have already classified those molecular patterns. They are lipopolysaccharides, peptidoglycans, nucleic acids, and single-stranded and double-stranded RNA and flagellin. Based on these, those immune cells respond and they bring about innate mechanism. And that innate mechanism of protection is the third line of defense. Okay, third line of defense. And in the third line of defense, the extension of the third line of defense is acquired immunity. The extension of third line of defense, the same extends further, is acquired immunity. Now, what is this acquired immunity? More details of that I'm going to talk about tomorrow, but I'll give the introduction on that acquired immunity. Acquired immunity is also called as adaptive immunity. And it starts with the innate immunity, which leads to acquired immunity. If the innate immunity is good, acquired immunity is also going to be good. If the innate immunity is bad, acquired immunity is also going to be bad. And all this is determined by the genetic constitution of the individual. How he is born with, or how she is born with. All this mechanism is genetically controlled and dependent on the genome of the individual. Therefore, please remember, the response shown by one individual against the pathogen need not be the same as the other individual. It varies from individual to individual. Therefore, immune response is individual specific. In some, it is mild. In some, it is overwhelming. And in some, it is very, very severe. It is all in the genes. How an individual responds is based on his genes. Therefore, whenever an individual is going to be treated against any pathogenic infection, it has to be individual specific treatment based on the individual's response. You cannot generalize, but generally what is practiced? The general treatment practice is used to treat everyone in the same manner, which is not a good procedure. because some will respond positively and some will not respond. Therefore, it is always better to do a proper investigation and then decide about the treatment protocol for an individual. So, about the acquired immunity, individual acquires immunity during his lifetime by the clinical, subclinical infections of pathogens, but may not develop acquired immunity for some pathogens which are deadly, which cause death within 48 hours. Therefore, against those pathogens which cause death within 48 hours, the individual should be protected against those pathogens, okay, through artificial means. And that artificial means is nothing but vaccination. You have heard about vaccines, and vaccines are used for giving acquired immunity to the person, to give protection to the person against those deadly pathogens. And that is what they are talking about now, the vaccines against coronavirus also. To give protection to the person, irrespective of the person who may recover, who may not recover. If you protect him, the person will not get the infection to such an extent. Fine. So acquired immunity is adaptive immunity. It is called as why? Because you adapt and you overcome the disease, and your immune system comes over it, recognizes it, produces the immune system responsible for cell mediated immune response, where T cells are going to be activated and the B cells are going to be activated for production of antibody, and you will produce both these systems properly against that pathogen, and you overcome the infection. With the result, you survive and you adapt to it. Once you adapt to it, next time if the same pathogen has infected you, you will respond very quickly. They were saying here that when the coronavirus infection takes place, minimum 14 days are required to detect the antibody. Yes, it is not only against coronavirus, it is against almost all pathogens. Please remember, it takes 48 hours for a person to get immunologically activated and start producing the active T cells, T lymphocytes, and the 2 T helper cells to come to your defense and start producing cytokines to activate the other part of the immune system. And it will take minimum 1 week to start producing the antibody by the B cells. So therefore, whenever you employ a technique to detect the antibodies in a person, if you use the technique 1 week, within 1 week of the infection, you will not be able to detect the antibodies. And you will give a wrong diagnosis stating that the person is not infected, but you are looking for a wrong thing at the wrong time. If you're using the kit to detect the antibody, that kit should be used for detection of the antibody for the infection only after 1 week of the infection. The antibodies will start making their presence in the plasma after 7 days. But before that, if you do the investigation for the antigens, at that time there will be very high load of antigen in the blood of the patient who is developing the symptom. And if you have the mechanism of recognition of that antigen using proper tools, you can recognize the antigens without fail. But if you employ the technique of recognition of antigen after 14 or 20 days, then you may fail to recognize the antigen. Why? Because that antigen has been destroyed by your antibodies. So therefore, whatever the technique that you employ for the recognition, it should be appropriate at the right time. Just because the kit and technique is available, you can't apply the technique as and when you please. Antibody detection technique only after 7 days of infection. After that, and up to 1 month you can detect that. Antibodies will be present in the plasma of a person for 1 month. High titer antibodies will be there, and then they will slowly start decreasing. Antibody detection can be done after 1 week of infection. But antigen recognition should be done within 10 days. After that, if you try to recognize it, possibility that the antigen will have disappeared from the body because of the immune response of the person. So the acquired immune response comes later as a supporting immune response to the innate immune mechanism that you have, and the acquired immune response lives lifelong with you until you die. Only thing is, the immune response slows down with the age, but it doesn't disappear. But acquired immune response may disappear in a person who goes through the chemotherapy or radiation therapy for various treatments. And whenever a person is treated with chemotherapeutic drugs, the possibility is that the person may suffer causes immunodeficiency. And that time the person has to be given assistance, and that is called as passive immunity. And we will be talking about that passive immunity in acquired immune response in the later part of our discussion.